when we made the decision to start this new bioengineering program, we wanted to have it be very science-based. So the toxicology program decided to move into the School of Engineering and join us to try to think about molecular to systems level, all kind of things. What that did for the engineers like me is have us start thinking about problems in prevention and in toxicology, whereas a lot of us have been thinking about problems in medicine. So I became, at one point, a member of the Center for Environmental Health Sciences, and I think that has been, at MIT, a very effective center in bringing in people like me who may not have thought about toxicology, but now see it as an incredible frontier and incredibly important for all kinds of health reasons. I don't have it listed, but given some of the things here at this meeting, I should say I'm also a member of the MIT Cell Decision Processes Center. That's a cell signaling pathways, computational biology meets systems biology meets tissue engineering kind of endeavor, joint between MIT and Harvard Medical School. Peter Sorter and Doug Laufenberger head that. And so much of what I'm going to talk about is informed by thinking of how we can combine new kinds of physiological culture models that capture complex tissue physiology in ways that we might use it for computational toxicology. And I have very much a talk in the theme of really what are some overarching questions that we're trying to answer. So I'll start by laying a framework of something we think about every day. If there's a simple way to do something, if you can do it in the kind of 384 well plate or bigger with a single cell culture, it's always going to be easier and cheaper to do it that way. You may do it with primary cells, but it's always going to be easier and cheaper to do it with monocultures. So an overarching question we always think about is that the choice of the culture system has to be driven by what your question is. What exact facets of tissue physiology are relevant, and why would you need a co-culture in order to capture it? So for example, I showed liver. It's a system I work in a lot. They're not the only system. Where you have hepatocytes that we think of as carrying out most of the toxicology events. They die, they convert compounds, they die, but of course they interact with a number of other cell types in liver, notably endothelial cells, stellate cells that deposit matrix, and over time chronic exposure to compounds can induce fibrosis and cirrhosis, so you can have long-term events. Obviously interaction with immune system cells, either acutely or chronically, may contribute to these outcomes. And I show here blood flowing through the tissue, and as we move into tissue co-culture and organ cultures, these kind of effects would be perhaps very important. So we can start by asking always what facets of tissue physiology are relevant, what is the easiest and cheapest way to capture that. And so two reasons to go to more sophisticated cultures than just primary cultures is that you want to enhance cellular function. Maybe you cannot keep a differentiated hepatocyte function just by culturing it alone. Or, importantly, you get synergistic effects from cell-cell interactions that happen in the tissue. So, co-culture part one, what do monocultures capture and miss? And all the discussion today will be on primary cells. So I'm going to take one step up from the previous talk. We focus on primary cells, but we think a lot about whether we can actually model relevant physiological processes in monocultures. I have a little bit different take on the use of toxicology in drug development. And again, I'm going to use that as a model as well. I think some folks in the drug industry have picked up on this idea of dose response. And so I just show here as an example of work out of Pfizer, where they took over 300 drugs on the dealing list and used a combination of imaging assays, sublethal assays, to screen these and correlate with known in vivo results. And what they found here, much better statistics than maybe previous kinds of assays, a true positive rate of about 50 to 60%, false positive rate, very, very low false positive. And importantly, they identified many idiosyncratic toxicities. The focus of this particular study was to determine whether these kind of medium throughput screening, high information content would allow them to both parse mechanism and also identify idiosyncratic toxicity. So they did well, but not perfect. So there's a positive rate, but they're still missing. So this is very interesting because it suggests a very simple assay with a high information content from it, primary cells cultured in a simple way, 96-well plates can be useful. But we can ask whether the positive rate is deficient because of insufficient retention of hepatocyte function culture. It's known that these cells are horribly susceptible to loss of metabolic function culture, although those experiments were done very soon after cells were plated. So one reason that to go to a co-culture system, particularly for cells like hepatocytes, which are very have many, many complicated functions, is to improve metabolic function. And it's been known for many decades, going back to the pioneering work by Christian Guizon-Pulizo, that co-culturing hepatocytes with other cell types can allow them to retain function longer in culture. And with recent advances in biomaterials and micropatterning, this has been systematized, most notably by Sangeeta Bhatia's lab, and this is images from a paper in Nature Biotech showing that you can create micropattern cultures of hepatocytes in an environment of, in this case, mouse fibroblast cell line that tends to support their long-term differentiated function. And then, in fact, even out at many days of culture, you can capture accurately a few toxicities of a number of different compounds. Okay, so these have an advantage that you can keep them for many weeks in culture and potentially be susceptible to chronic exposure. Whether these can capture everything on the DAILY list, it's a tremendous undertaking to test all of those compounds. So I think these kind of technologies are emerging to fill those gaps. The way, now if we go back and say, what question are we asking? What is the physiology of idiosyncratic toxicity? It's helpful to think of whether a hepatocyte or hepatocyte plus mouse fibroblast culture would capture that. So if we go back and think about it, or look at the hypotheses that there's genetic polymorphisms, or immune response due to adducts form, but emerging in a lot of situations, particularly with changes in the population, is that there's a relationship with some kind of inflammatory insult, and that's been very much popularized or demonstrated in animal models, at least, by Bob Roth. And our own interest in this was sparked by a project joint with Amgen, who had a drug that ended up being idiosyncratic toxic by this mechanism. So as we know, in the normal physiology, your gut sits there and protects you from all the LPS. Not all of it, a little bit leaks out and reaches the liver. But when you take a drug under these circumstances, if it's not toxic, it sits here. But under events that may increase gut permeability, which may in fact be the drug, and this was the case with the Amgen compound, a lot more LPS goes to the liver, and then you get fatal kind of interaction between the cytokine-mediated events in the sinusoid and the drug, and that causes the liver to crash. And this, importantly, is not a linear combination of effects due to inflammation and drug. It's actually a synergistic combination. So one could ask, well, we could build a model of the liver. You know, we could try to build a model that incorporates all of these cellular elements to capture this. But is there a way we can try to parse it and actually build the cues in with just a hepatocyte culture? So again, I work in tissue engineering. I think a lot about building these complicated models. But if you can do it by capturing the relevant cues with just a monoculture, that's always going to probably be cheaper. So let's just talk a little bit about that, and then I'll come back to that. So together with Doug Laufenberger at MIT, again, as part of the Cell Decision Process Center, and this was in collaboration also with Pfizer, one can think about doing a high-throughput screening approach where your hepatocytes sit there and they're in an environment where they can get cues that are cytokines that can be released during an inflammatory event, and then the drug. So you have a comorbidity, and you can test different kinds of cell platforms. And importantly, where we're going is we're trying to link some of these events to intracellular signaling pathways. So there's a lot of measurement of kinase, multiplex measurements of kinase signalings and attempts to develop data-driven models of how these drugs act. And here we did just some very simple cell death assays, later validated a little bit with sublethal assays, just to test whether we could capture some of this idiosyncratic toxicity. So this is just one example to give you a flavor for how just this simple model can be enhanced by building in cues that you have inflammation. So here's the drug alone, and these are HepG2s, but we did it with primary human and primary rat as well in this paper. So here's, and this again, just LDH release. We did other assays of toxicity as well. And so this is just the drug, okay? And now if we add the cytokine mix, mix, and we tested a whole bunch of cytokine mixes, and there's a rationale for what we did laid out in the paper. You can see there's a tiny bit of increase, there's a tiny bit of death that comes just from cytokines. We've got no drug, cytokines do this, and if we simply add in a linear fashion that amount of death to the drug curve, we get the red line. But in fact, if you actually give the cytokine mix, as shown here, along with the drug at these concentrations, so you give the cytokine mix, in fact, you get a synergistic response. You see this gray area is where you're getting synergistic from just these simple assays. Okay, so we went in and did a screen of primary human hepatocytes, 90 drugs on the daily list, different categories, and you can see there's again a rationale for how we pick the multi-dose concentrations, but you can see here every drug for which there's pink below this line has some idiosyncratic toxicity. Okay? And so we went in then and have done a lot of measurements of signaling and relating those toxicities to signaling pathways in hopes that we can have simple screens that help us pull out idiosyncratic toxicity. But in fact, we're still not all the way there. Even though this helps, it doesn't do everything. And so we continue to ask whether hepatocyte-centric models will capture all toxicities, even if we try to replicate these other factors. So that brings us to the concept that we want to really maybe we can screen things in these compounds, catch a lot of things, but at some level then we have to go to a lower throughput assay, which has more complexity and captures fastest tissue. Now, there's been available for many, many years availability to do liver slice cultures. It's done not super widespread. It's somewhat of an art to create them, but you can do fast off preparation from almost any organism, even very thin slices. They tend to be viable only for short term. There's a very large number of papers using liver slices showing that they're incredibly physiologically responsive. For example, it's a really nice study showing zonal toxicity of bile acids in cultures developing over 2 days. But we still have questions of longer-term maintenance of these higher throughput cultures. And again, slices can be very useful to work with if there's a little bit easier way to do it. And so work out of Kenny Groothuis' lab in the Netherlands, together with the microfabrication experts in the report, has now started taking slice culture to the next level, using microfluidic systems. And in particular, this is a really great idea because you contain the slice in a very, very small microfluidic system, allowing very controlled perfusion around the tissue of precise microenvironment in terms of culture medium around the tissue. You can also rapidly and in a very controlled way change what's in the medium, so it holds compounds, exposed compounds for days or hours, and change it. Very well controlled microenvironmental conditions. This is just a little picture of the assay. And so these kinds of things are now starting to come out to make these kinds of cultures more standard and perhaps reliable and accessible for toxicity assays. However, these cultures can be maintained longer, still probably not long enough for really high throughput assays. I'm going to use that as a jumping off point to a little philosophy on where we are with microfluidic devices. There's an explosion of activity in developing methodologies to culture cells in microfluidic devices, complex cultures. A paper that got a lot of attention, generated an incredible amount of enthusiasm out of Steve Quake's lab. It's one of the real leaders in developing these. Is a highly multiplex way of measuring hundreds of different conditions. Here showing stem cell differentiation, how you get multiple different cytokine growth factor matrix effects in a multiplex way on stem cell differentiation. And so there's certainly the potential to do these kinds of things with primary cells, even with matrix and growth factor control, but we're still maybe not quite at the point it's generalizable. And there's a nice review in a recent Nature Methods on the sort of state of the art of microfluidics for these kinds of assays. And it really highlights the need to move things out of individual labs like Steve Quake's lab for such an experiment is a tour de force, as noted here by the author of this paper, and really move it into a commercial platform. And companies like Fluidine and Cellasics are now starting to do that, so they're developing really commercial platform technologies that can be made more accessible to the community. We're a ways from there, But as we approach that in parallel, there's some really great things going on in the microfluidics community, developing ways that can be incorporated into these multiplex assays. For example, Lee's lab at Berkeley is a leader in developing complex 3D cultured microfluidics. He's also affiliated with Computational, who's again trying to translate some of these things into practice. This is 3D liver culture, which allows you to have gradients and again fast exchange of nutrients, very well controlled microenvironment, and with microfluidics you can ramp up and ramp down factors in the media. There are emerging microfluidic systems to model interactive organ systems. Some of you may have seen this from the Jurel system. This actually came out of Mike Schuler's lab at Cornell. Again, he has long been a leader in pharmacokinetic modeling. He's a chemical engineer and has been using microfluidic systems to make interacting organ systems. Now, his organ systems are 3D, but they're right now based on cell lines. But it's really interesting emerging technology. This is, again, an example of this, where you can see toxicities on a target organ, like marrow or a tumor, based on metabolism and habits of the organ. So these things are, again, still in the realm of not quite ready for general use, but emerging. I work in the area of 3D patterning, and I can attest it remains incredibly challenging. Patterning in 2 dimensions is easy. You teach a high school student to do it. Patterning in 3 dimensions remains incredibly challenging. And we're faced with issues of resolution. So, for example, organ printing has gotten a lot of press, but the resolution is really not great. It's 300 microns, where it's not at the cell level yet. George Whiteside's lab has been experimenting with modular organ assembly using microfluidics to make little tissue constructs here. These are just 3T3 cells, but again, you see the resolution is on the order of hundreds of microns. Roger Cam's lab, in collaboration with my lab, has been developing microfluidic systems. So let's put hepatocytes here, endothelial cells here to try to across a gel matrix. This is about 200 microns to look at angiogenesis into this. So there are a lot of approaches. Unfortunately, most of the 3D fabrication methods rely on lithography that requires UV irradiation. UV irradiation, some cells certainly live through it, but it always will alter the phenotype of the cells. So we're still a lot of activity going on, no really facile way to do this yet. Further, most 3D phenotypes, as we get into 3D, are governed by the mechanics of both matrix, extracellular matrix, and flow. And flow, again, is an incredibly difficult thing to control in 3 dimensions. So just, again, philosophically, a few things that could come in to influence even our single-cell cultures if we think about cues that regulate environment, simply thinking about the substrate we culture cells on and its mechanical properties in addition to whatever composition of matrix. Very seminal paper by Dennis Disher's group illustrated for mesenchymal stem cells, if you simply change the elastic modulus of the substrate from very soft to much stiffer, and these are along the range of brain to muscle to bone mechanical properties, you can in fact drive differentiation of these mesenchymal stem cells even if you don't add components to the medium. So there's matrix effects, and this certainly is also true activation of hepatic stellate cells. As you increase the stiffness of the matrix, you drive an activated phenotype like you have in cirrhosis and work out of Becking-Wells pen. And again, getting into 3D, you get incredibly more complex because you need flow to nourish the tissue, but flow also exerts mechanical stresses that can greatly influence cell function. Simple example here, monolayer of endothelial cells growing on top of the gel. We can put a liquid on top and let it flow through into an underlying gel. A higher height of liquid creates a greater flow velocity. And if we look at just umbilical vein, these are just endothelial cells, no flow, and just very slow flow, like you would have an interstitial flow. This isn't even what you would have through the vessel, along the vessel, but actually through the wall of the vessel. You can see that if we look at the effect on cell number, there's a velocity where you get an enormous increase in proliferation. And this is consistent with the velocity that you actually have moving from capillary to vein. And there's a capillary morphogenesis associated with this. In our own work, so in patterning individual cells is complex in 3D. In our own work, we've been, and this is work inspired a lot by working with Yvonne and his challenges, and particularly in creating inflammation models. is trying to create 3D liver models, not from slices, but dissociate the cells and recombine the cells physiologically. And here we've just tried to mix the relevant cell types into a 3D scaffold where they can, to some extent, self-organize in tissue-like structures. So the idea here is that we have a 3D scaffold which is holes in a silicon or polymer wafer coated with matrix, seed cells in here. They adhere to the walls of these channels. Initially, they're supported by a filter that distributes flow. Culture medium can flow through at the cells initially here. Culture medium flows through and nourishes the cells and provides flow. And so this is an initial imaging version of this reactor that we made, and you can foster histotypic structure in collaboration with Ivan and supported in part by NIEHS. We've adapted this methodology to a multi-well plate array. We're right now in a 12-well plate, captures the essential features. The cells live here. There's a continuous flow of fluid generated by a mini peristaltic pump, microfluidic peristaltic pump. So we circulate fluid around and around, but it's now in a very facile kind of multi-well plate format, again, capturing the essential features of making these 3D tissue constructs. And here, what we've been very interested in is the effects of flow on tissue function. And so, for example, if we look at endothelial cell function, and here this is a 3D composite focal image of cells growing in a channel, and this is 2 weeks after cells were initiated in culture, so long time scale in the realm of liver cell culture. And what we're staining for are sinusoidal endothelial cells, which are incredibly fragile in culture. At low flow conditions, they're maintained. At high flow conditions, they disappear, so they're red. And this is partly because there's a proliferation of stellate cells here, and they've had secretion of TGF-beta. So I'll finish up by coming back to this inflammation model of idiosyncratic toxicity, because these 3D models may be, we haven't proved it, but we're starting to see that they may be a way that we can capture these effects. Now, this slide in the conversion from MAP to PC, the data didn't come through, but the title is what we would show you, that if we have co-cultures of hepatocytes plus non-parenchymal cells, particularly we add in physiological ratios of these, they respond to inflammatory signals, particularly LPS stimulation. They can do it repeatedly over many days. So you can stimulate, let them rest, re-stimulate, and then respond again. So here we are using this to examine these idiosyncratic toxicities and how they may be mediated by inflammation. So the model that we actually have worked with Pfizer. We've translated our system into Pfizer. We're working with other companies. There's incredible interest in the drug industry. So I think these two will be, and I think the kinds of models we develop are appropriate for toxicology testing. What is moving into the commercially available realm is already available, as with many things, with regenerative medicine. Very straightforward models. Still, to get them to work robustly is a major challenge. Tissue engineering skin was the first thing to move into the clinic, and likewise skin has moved into the realm of toxicology testing with MapTech's various models. This is one that they recently validated across several labs to do a microfluidic sampling. So little steps starting to be commercially available. Again, the overriding, you have to frame your overriding question, and the level of complexity that you'll use will be related to that. So just a few parting thoughts. There's incredible activity now fusing biomaterials and microfluidics. We're learning a lot about how to engineer the cell environment to get long-term maintenance and also interactive cell systems. There's always going to be a balance between high throughput or medium throughput and high information content. And the commercialization is going to be really needed to spread these platforms to the state that we get to use them. And so the whole role of the stem cell compartment in tissues is also one very important, I think, particularly for toxicology. We and others have looked at this batch issue and found that there are ways to handle it. For example, in the signaling pathway network, we did a screen, a toxicology screen on cells from a particular donor and developed a model for how the signaling pathways would predict toxicology and use various computational models to do that, and then use inhibitors with a different donor to show that you could get that the predictions of the model actually held up. And that was actually just even with, in the end, 3 donors. So very different donors, actually, different properties of donors. So I think there's a lot of effort to try to look at common effector pathways and ways that you could correct for that. But how the whole genetics and epigenetics plays out, I think, is still emerging. and what role the genetics plays in the toxicology versus some of these other factors that I outlined.